everyone, welcome to my channel Pure Biology, and today I'll come with another video, that is the RNA editing. And before going to this topic, I want to say thank you to all of you who have already subscribed my channel and support me a lot for the growth of this channel. And I request you, the newcomers, if this channel is helpful to you, so please you just share and subscribe this channel. And of course you just press the bell icon after the subscription or after subscribing the channel for getting all the notifications when I upload my video. You can get all that. Okay, so let's start the RNA editing. What is the RNA editing? RNA editing means the editing of RNA. If the RNA size is very small, then the RNA must be extended, or if the RNA is extended much, then it is obviously deleted some of the nucleotide bases. So this is called the RNA editing. Okay, and this RNA editing is seen in everywhere. That means this is the widespread, and it is seen in the all eukaryotes, like the yeast to human. Okay. And there are two types of editing process. One is the specific base modification editing, and another is the insertion deletion editing. So site-specific base editing includes the nucleotide transition or transversion in RNA. Okay, nucleotide transition or transversion in RNA. And generally two common types of editing processes seen. Generally C to U, U means uracil, and another is A to I, that means inosine. Okay, I for inosine. So this C to U transformation, it is seen in the human mRNA. Okay, and this occurs with the help of this protein. This is called the apolipoprotein B. Okay, and the gene for the protein codes for 4563 amino acid polypeptides, and this gene is ApoB100, that synthesized in the liver cell. Okay, and which is secreted in the bloodstream. Another protein, this is the APO848 protein. This is made by the intestinal cells. So here the two examples are there. The one is the ApoB100, that is synthesized. It is one kind of protein. It is synthesized in the liver cell. And another protein, this is called the APO848, this protein, and this is made by the intestinal cells. Okay. So in case of intestinal cells, the mRNA is modified by the deamination. Okay, here this processing occurs by the deamination process or with the help of deaminase enzymes. Clear? So here in case of intestinal cells, this the mRNA that is modified by the process of cytidine deaminase, and the process is deamination, but enzyme is the cytidine deaminase. And here the deamination occurs of what? The deamination occurs of the cytosine. Okay, and that cytosine is converted to uracil. Cytosine is converted to uracil. Okay, so next thing is that these changes in that case where the cytosine changes to uracil, what happened after changing of cytosine to uracil? You can see that codon CAA, this codon CAA, this is specifying for the glutamine. Okay, this is specifying for the glutamine. But when this changes or conversion occur, then this CAA codon that into UAA codon, and which causes the translation stop. Clear? What is saying? Again I repeat, the changes a CAA codon that present in this position, that specifying the glutamine, and that into UAA, which codon causes the translation stop. Clear? So I think you can understand properly that what happening, this, if cytosine is present, that means the codon is CAA, and here the glutamine is specified, and if that is converted to uracil, then codon UAA, that responsible for the translation to stop. Clear? Next one is the another example, that is the A to I editing. A to I editing means I for inosine, and this A to I editing, deamination of adenosine that results in the formation of inosine and carried out by the adenosine deaminases. So here the conversion of adenosine to I for inosine, and that is occurred by the adenosine deaminases. Clear? And this inosine that behaves like guanosine and pairs with cytosine. Okay, so this is the example of RNA editing process. One example, that is the site specific base modification. The second one is the insertion and the deletion editing, and this is occurred in mitochondrial RNA and in where? In the protozoans. Here the nucleotides that are inserted into or deleted. So here nucleotides may be inserted or deleted. And obviously here the ligation process is involved just after insertion or the deletion. Clear? And this is one enzyme is very helpful for causing or for creating this process, that is the 20-S aditosome. 20-S aditosome. Clear? And the sites of the pre-mRNA to be edited complementary to edited RNA sequences. So what is saying that this is the pre-mRNA, and the after editing, that edited RNA, they are complementary to each other. Okay, and this edited RNA sequence, and when the complementary to the pre-mRNA, this is another name is the guide RNAs, or you can say the gRNA. And this gRNA has the three sites. One is the 5' region, another is the central domain region, and third one is the 3' end region. The 5' region, that is complementary pre-mRNA, and central region which contains some information may be inserted or deleted, and 3' end, that is characterized by the poly-U tail. So this is very short or brief description of the editing process, and I think this editing process is involved in the RNA processing, and where the another two processes also involved there, that is the polyadenylation and the 5' capping. These are already described by me and uploaded in my channel, so you just check out that one, and then you can understand this RNA editing process. Now I will explain about the spliceosome, what is the splicing of RNA occur, and this is explained in the next video, so you just check out. Okay, thank you. Sorry, for another thing I have to explain that this editing process that occurs by the 4 stages, and this occurs in 3' to 5' direction. 4 stages mean the first one is the gRNA or mRNA, that duplex formation occur. Second one is the cleavage of mRNA at the target site, and third one is the addition or the deletion in the mRNA, and the fourth one is the religation of 2 fragments of newly edited mRNA. So these are the 4 stages or processes that including or that involved in this RNA editing process, okay, of the gRNA. And here depending on the information, the insertion of uridylates or deletion of uridylates that occur, and if the insertion of uridylates that occur, the enzyme involved, this is known as the uridyl transferase, and if the deletion is needed, so here the enzyme is uridyl exonuclease. Okay, so now RNA editing process is completed. I thought that this part is not there, so but next I checked that this part is also included with this. Okay, so I have to explain that. Okay, you just continue with my channel.